Hi everyone, my name is Ana Pastrana, and today, together with Johanna Castillo and Lydia Tymon, we are going to talk about sanitation practices for nurseries and greenhouses. So first of all is like why we should care about pests and diseases. So we should care about pests and diseases because diseases and pests lower the yield of our plants, change their susceptibility to other factors, also could change the performance, taste, and appearance, which is very important in nurseries. Ultimately, because they cause plant death, so we cannot sell our plants. So today I'm going to focus in more general methods. My colleagues Johanna and Lydia are going to focus a little more on them. So today I'm going to talk about how to clean facilities, use certified plants, keep the ground of our nurseries and greenhouse clean, and how to clean our equipment. So first I'm going to talk about cleaning our facilities. So when should we clean our facilities? So we should clean them as soon as possible. It's better to clean them preventative than curative. And also the best way to clean our greenhouses are normally when they are totally empty. So the best time to clean our greenhouses is between crops. So once the greenhouse is empty, then is when we should clean it before bringing in new plants. So how we should clean our facilities? So the first thing that we had to do is removing all the plants. So first we are going to dry our plants, trash them, and remove all the drippers, all the substrate, and collect all the trash that we have in our greenhouse. Then we are going to also remove all the surface debris because these organic residues from plants or from growing media can reduce the effectiveness of the disinfectants that we are going to use. And they can also allow pathogens to survive longer. Like, for example, if we have weeds around or crop debris, they are going to allow pathogens to survive longer. And one important thing is that when we clean up all these plant debris, it's very important that we don't compost them next to our production site. So it's better that we trash and kill them properly, like far away, because I have been seeing a lot of like people that clean their tunnels or the greenhouses, and they compost the debris and the trash next to that greenhouse, which is not the best because pathogens can still survive in those compost piles and make it through to enter again in our greenhouse. So it's very important to compost them far away. So once our greenhouse is clean, we don't have any dirt or plant material, what is best is to try to wash all the surfaces with soap and water. So watering the surface with soap and water is going to make us more sure that we don't have anything remaining that is going to inactivate the sterilizing agent to follow. So once we clean with soap and water, we are going to rinse off anything that is remaining, and then we are going to allow the soap and water to dry. Once it's dry, is when we are going to apply the sterilizing agent that we choose. So then is when this sterilizing agent is going to be the most effective. So we are going to apply the sterilizing agent, and we are going to allow this disinfectant to dry again. And still, if there is any residual debris from the cleaning area, we are going to remove them. So once we have our greenhouse clean and disinfected, is when we are going to bring plants again to the inside. So the plants that we should bring to the inside of our greenhouse or facilities, it's better that we buy always certified plants, because by buying certified plants, we are sure that they are clean. So so we had to buy plants from nurseries or like areas that are like we trust on those sources. So even when we trust on the source of the material, it's always good to scout the new material and see if the new material is coming with any leaf spots or any virus symptoms that it may make us suspicious of this material. So normally, if we see any kind of symptoms, it's better to reject those plants. If we don't know 100%, because sometimes at this stage, a lot of diseases are asymptomatic, they don't show any symptoms. So sometimes it's better to place this new material in a quarantine area and wait around 2 months to see if any disease show up in those 2 months. Once 2 months have happened and we are sure that any disease are coming with this new material, is fine to put other plants around them. So after that, I'm going to talk about how to keep the ground clean, because there are a lot of nurseries that they don't have, like, as I showed before, they don't have tunnels or greenhouses, and they are like just benches on the outside. So in this kind of nurseries and greenhouses that doesn't have any big equipment, just the pots, it's important always to try to keep these pots and plants out of the ground as much as possible, because if we have them in direct contact with the ground, normally there are a lot of like diseases that can splash on them, like for example Phytophthora or Pythiums. But if we keep the ground clean and put these pots in benches, we are going to avoid these pathogens that can come from the soil or from the splash. Another way to keep to minimize the splash is to try to avoid to avoid these puddles next to our plants, and try to collect the standing water and improve drainage. As I said before, it's very important to keep the ground clean of this waste. This is a picture that you can find in many greenhouses, where they collect all the dirt material and compost them and put these piles of pots dirty next to other new plants. So it's better to eliminate this trash around our facilities and try to trash them far away and keep our facility clean from this trash. And the last thing I'm going to talk about is equipment. A lot of diseases move from plant to plant through the equipment, through pruners, even through the people working in them. So the best way to try to avoid the dispersal of diseases is to try to always work from clean to dirty houses. Like for example, if we know there is like a house that is we have new plants, we have plants that needs to be pretty clean from diseases, it's better to work from those ones first, use the ones that we already know that have diseases to work at the end of the day. I know that is not always possible, but it will be like super useful for preventing diseases if we have if we have several houses and we can have like a set of tools for each house, so we don't share diseases between houses. But if this is not possible, a good practice is to try to decontaminate tools from house to house or from site to site. Many times it will be also good to be able to use gloves of, for example, food covers when we know that we can have a soil-borne pathogen. It's good to have food covers to share between different sites or different houses and try to avoid cross-contamination. The first thing to decontaminate our tools is to try to take care of our tools as much as possible. Try to avoid nicks, scratches, and ridge on these tools, because these things create like pockets, and those pockets can harbor microorganisms and pathogens that can move from plant to plant. So sometimes these tools also get dirty from plant sap. It's important to also eliminate that plant sap between houses at least. So some of the cleaning methods that we can use is just soap and water. Sometimes it's useful. Also high pressure water or soaking hot water are going to be better. And if we wanted to remove this latex, this milky sap, we can also use paint thinner. Another good way to decontaminate our tools is like to apply a flame. So in some cases, this is pretty simple. It could be done fast. In some seconds, we can try to flame our tool and try to eliminate any pathogens that can be harboring in the tools. Boiling water is also a cheap alternative if we don't have steam equipment. Of course, steam sterilization is the best to try to sterilize large equipment like carts, machinery, and pots, and trays. Here there are like several disinfectant agents that are in the market that are suitable for being used either to decontaminate facilities or to decontaminate tools. Now I'm going to let my colleague Johanna to share her presentation and follow me.
Okay, so, and it's in presentation mode, correct? Perfect. Okay, so I will, as Anna, have some general overview of sanitation practices that we have in greenhouse and nurseries. I will talk about informed sanitation practices based on a case study of a research project that I conducted working with vegetable transplant nurseries. So a few years ago, what we did is we went to different nursery facilities and we're looking into like which were the critical control points in the California transplant production. What we mean by critical control points is steps of processes in the production of plants that may pose a greater risk for pathogen contamination. And so we went and we sampled the trays, we sampled the benches, we sampled substrate, we sampled water, and we assess, and of course, plant samples. And also we look into like microbes present in these surfaces. And so that is the results based on that we'll be sharing. So one of the main critical control points that we identified were benches. So we learned benches are pretty diverse. We can have in here these benches made of wood at a pretty close distance from the ground or basically in the ground. And also we have benches made of metal that are raised from the ground and that are on paved or plastic covered floors. And so based on the discrepancies or the difference that we saw on the benches we sampled, we established certain categories. So one, we have benches based on height. So we have low, medium, or high, and you can see the distance in the different categories that we selected. So low was from 2 to 6 inches from the ground, medium to 20 over 20 inches, and height over 28 inches. In materials, we had benches that were made of wood or metal, or only metal. And for disinfestation methods, different facilities applied no treatment, or some applied hydrogen peroxide, chlorine, or quaternary ammonium. And so based on this, we measured the microbial loads. In this case, what we'll be showing now is the fungal propagules that we collected from these surfaces. So what you will see in this figure on the y-axis are the fungal CFUs, or colony forming units, that is an equivalent to propagules. And so what we have is that benches that were at a lowest distance from the ground, from 2 to 6 inches, harbor at least 86% more fungal propagules than benches that were at least at 20 inches from the ground. And so basically, the closer the benches are from the ground, the higher chances that by water splash, as Anna mentioned, that you will get microbes present in the surface. Then we're looking into bench materials, and we found that benches that were made of wood harbor at least 93% more fungal propagules than benches made of metal or a mix of wood and metal. The reason for that is that wood could be a material where microbes can't harbor and hibernate, as compared with metal that is less likely that microbes can overwinter in those surfaces. And then we look at disinfestation treatments that were in place at the different nurseries that we sampled. And overall, we found that benches that were sanitized with quaternary ammonium harbor 99% less microbes than the ones that had no treatment. Or for one of the nurseries, we found high loads of microbes that were treated with hydrogen peroxide. And all this was based in a confounding effect of besides the woods and the height. And so we don't have effect per se for disinfectant, and I will go to that in a minute. So as I mentioned, as there was a strong confounding effect into what made a bench, so as like the height, the disinfectant, the material. And so when we did the statistical analysis, we combined all those different characteristics of a bench, and we found that the benches that were at a lowest distance from the ground, made of wood, harbor significantly higher fungal loads than the benches, for instance, that were made of metal at a highest distance from the ground, that was 28 inches, and have the floors like with paved or plastic cover. So based on that, best management practices that we can propose in regards of benches is that raised benches at least 20 inches that made of metal or a combination of metal and wood significantly reduce the presence of microbial loads. It doesn't mean that all the microbe loads that we recover are pathogens, but the more microbes you have, it increases the likelihood that some pathogens may be present there. And then surface disinfectant, based on what growers were applying, we identified that quaternary ammonium or chlorine were most effective methods. But a question that we have is, are evaluating only the effect of disinfectant, is not like in combination with material or in combination with plant height, what the surface disinfectants were more effective. So to answer that question, we performed some experiments in the greenhouses here at UC Davis. Sophia Acker, a former member of the lab, inoculated some metal benches with Botrytis cinerea, which is a pretty common fungal pathogen present in California nurseries. And then we applied treatments of quaternary ammonium, sodium hypochlorite, hydrogen peroxide, and peracetic acid, water, and no treatment. And so after we applied the sanitation treatment in those different like squares that you see there in the picture, we then went with swabs and evaluate the treatment efficacy by counting colony forming units present in the surface that we recover. So overall, what we found is in this figure, what you can have in the y-axis is the average Botrytis colonies that we recover after sanitation treatment. And in the x-axis, you see the different sanitizers that we have. And so what we found is that all sanitizers are very effective at eliminating microbial loads, in this case Botrytis, as compared to do nothing. This is the highest bar that we have here. But what we have is that chlorine treatments and quaternary ammonium were very effective at significantly reducing Botrytis propagules from the benches. Then we did these experiments many times as we needed to have reproducible results to provide recommendations. And in this case that we have, what we found again is that all treatments were very effective at eliminating botrytis than in this case the water treatment was higher or than doing nothing. But we have in here that chlorine was the treatment that was most significant effective at eliminating loads and was more significant than just applying water. And we found that hydrogen peroxide and quaternary ammonium were significantly more effective than only applying water, but in this case, but no treatment. And so overall, after many runs that we did with these experiments, what we found is that chlorine treatments at 5,000 parts per million, and also in some cases quaternary ammonium, were the most effective sanitizers at eliminating botrytis cinerea inoculum. One of the challenges of using chlorine is that it can corrode metal surfaces. So a recommendation we have is to rinse with water minutes after the chlorine has been applied. Also, it's important to know that chlorine can become deactivated if it's left for a long time. So if once you make the chlorine solution, make sure to prepare it frequently. Bleach can degrade it with light and at high temperatures over 75 Fahrenheit degrees. And also it can be degraded in contact with organic matter. So following up what Anna mentioned, that's why it's pretty important to clean first and remove any plant debris, soil, or organic matter after applying your sanitizer. This is an example of resources we have in our AIR website. You can find a link there of the different examples of calculations for obtaining a 5,000 part per million of available chlorine and how to build the dilution. And you can find that at AIR Nursery at UC Davis at EDU in the chemical sanitizers part. So now moving on with the different critical control points, we identified a vegetable transplant nursery. The second point was trays. All nurseries reuse their trays between crops, but all nurseries steam sterilize their trays. And so you could see the different temperatures, most range from 120 to 180 Fahrenheit, and the duration of the sterilization goes from 45 to 120 minutes. Overall, that we found is that steam sterilization is a pretty effective treatment at eliminating fungal and bacterial loads. In this figure, you could see in the y-axis, the blue bars represent the fungal propagules from dirty trays, and the orange lines that you cannot see much are the loads of clean sterilized trays. This is for fungal, and this is the case for bacteria. So overall, what our experiment told us is that steam sterilization is able to eliminate fungal and bacterial loads from 95 to 97% compared with dirty trays. But then what happens after the trays are steam sterilized? That we know is very effective. All nurseries do it. All nurseries should keep doing it. But then it comes with the tray storage. So in many cases, for the case of vegetable transplant production, the trays are stored exposed to the environment in loading lots. And so basically what we have in this figure is the measure of fungal loads in dirty trays, which is the blue one, and then the fungal loads of clean sterilized trays that have been stored outside. So that's we call all sterilized trays. And what we found is that they're not statistically different. So basically, if you have steam sterilize your trays and then you leave them exposed to the environment, you are losing all the great work you did at eliminating microbes from that tray as they are exposed to the environment and then accumulating in microbes. And then if we compare the loads with new steam sterilized trays, you can barely see anything because steam sterilization is very effective at eliminating trays. So basically what this is telling us is after steam sterilizing your trays, you should store them in an enclosed facility or at least cover them with practice to keep your trays clean and prevent microbes from reaccumulating your clean trays. So wrapping up in my section, based on the work we did, the best management practices regarding sanitation that we can propose is that benches should be raised at least 20 inches from the ground. ideally made of metal or a combination of metal and wood to prevent the presence of microbes, and the surface disinfectant that are most effective for chlorine and quaternary ammonium. In terms of trays, tray sterilization is very effective at eliminating microbial loads from dirty trays, but definitely after the trays are stored, an adequate storage of these trays should be taken in place to maintain your tray surfaces clean. And with that, I will let Lydia continue with this part of the presentation, and we'll come with questions afterwards.
Tymon, and I'm the Environmental Horticulture Advisor for San Bernardino and Riverside counties. And so today we're going to be talking about viruses in the greenhouses and nurseries, talking a little bit about their transmission and the role of sanitation in their management. So first of all, I wanted to define what a virus is. According to the American Phytopathological Society, a virus is defined as a submicroscopic intracellular obligate parasite that consists of a core of genetic material, which can be either single-stranded RNA or double-stranded DNA, that's usually surrounded by a protein coat or a capsid. And viruses can take on a number of different shapes. They can be spherical, they can be rod-like, they can also be helical or have a corkscrew sort of form, or they can take on the shape of a phage. Down at the bottom is a bacteriophage, and these are viruses that specifically infect bacterial cells. And viruses can either be non-enveloped, where they consist of just that capsid and the genetic material, or they can be enveloped, where the capsid and the genetic material is surrounded by a lipid or fatty layer. And this is going to come into play a little bit later with sanitation. And so to the left here, what we see is an image of a spherical virus. This is petunia vein clearing virus, and you can see the symptoms on the petunia leaves down below. So there's been a lifelong debate on whether or not viruses are living organisms, and technically they are not. The reason being that they don't really have the machinery for replication, for metabolism, and they really need to integrate into their host cells in order to perform those functions. And unlike other plant pathogens like nematodes, like fungi and bacteria, viruses lack the machinery and the capacity to move around on their own. And so in order to move from host to host or location to location, they really need to have a vector. And we're familiar with all of these vectors. They can be insects and mites, which are in the greenhouses and throughout nursery production. They can be nematodes, and we're not going to talk about nematodes today, but those are extremely relevant in field situations. But they can also be contaminated tools, humans with the human to plant transmission, and also viruses can be seed-borne. So in talking about insect vectors, we're all very familiar with them. The top four most common insect vectors for virus are aphids, whiteflies, thrips, and leafhoppers. So very problematic in our nurseries and greenhouses. And the reason for that is that they all have one thing in common, which is that piercing, sucking mouth part. And so if you look to the right on that enlarged head of the aphid, you can see that the aphid has inserted its stylet, which is sort of like a hypodermic needle, right into the plant tissue, which is a great way to transmit virus from the insect vector into the plant host. And so when we're talking about transmission, transmission depends on very specific virus-vector relationships. And this leads us into the discussion of persistence. And so persistence is essentially the time period with which the virus particles are associated with the vector. And persistence can be either non-persistent, viruses can be semi-persistent, they can be persistent circulative, or they can be persistent propagative. And so we're going to cover those next. So looking at virus persistence and transmission here in this chart, we have transmission, the site of virus retention, replication within the vector, the acquisition period, which is the time period it takes for the virus to be accumulated into the vector, and transmission time. So how long does it take for the vector to move the virus particles from itself into the plant host, and then particle retention within the insect vector. So when we're looking at non-persistent viruses, the site of virus retention is primarily in the stylet or that mouth part of the aphid or whitefly or what have you. There is no replication of the virus within the vector, and it only takes a very short period of time, just a few seconds, for the virus to be acquired by the insect pest. It's also very so for the virus particles to move back into the host, and the particle retention within the vector is only minutes to hours, so very, very short. For semi-persistent viruses, the site of virus retention is in the foregut. The viruses don't replicate within the vector. The acquisition period is minutes, so also a very short period of time. And then also minutes for acquisition. And the particle retention is just hours to days. For persistent circulative, the site of virus retention is within the hemolymph or the salivary glands, and there's no replication within that vector, and the acquisition is just hours, so a little bit longer than non-persistent and semi-persistent viruses. Transmission time is also hours, with the particle retention days to life. And then for persistent propagative, the site of virus retention is in the hemolymph and salivary glands. But this virus does replicate within the vector, and the acquisition period is similar to other persistent viruses, hours, with the transmission time hours to days, and then the particle retention is for the life of the vector. So how does that play into management and sanitation? Well, when we're talking about non-persistent and semi-persistent viruses, sanitation is absolutely critical for management. One reason for this is that you can't really rely on insecticides. So if you remember that acquisition and transmission time is seconds to minutes. And so it's really challenging for insecticides to have enough time to be effective against that vector. So what you have to rely on is sanitation practices. So that means removing infected plants or roguing, starting with virus-free propagation material or seed, and then also controlling your weeds and volunteer hosts, which serve as reservoirs where populations of insects can increase. With respect to semi-persistent viruses, there are not a lot of them that affect ornamental plants. However, those ornamental plants can serve as reservoirs for semi-persistent viruses that are pathogenic on other hosts. When we're talking about persistent viruses, insecticides are going to be a lot more effective for vector management, primarily due to the long acquisition and transmission time. So you have to remember we've got hours for those actions to occur. However, just because you have insecticides available, you can't forego your sanitation practices, which means that you should still be removing your plants and starting with the virus-free propagation material and controlling your weeds and volunteer hosts. What's a little bit different with persistent and propagative viruses is that you also need to make sure that you're controlling the immature life stages of your vector, because that virus is replicating within the vector, and those virus particles can be transmitted down to their progeny. So switching gears a little bit, sanitation for mechanical transmission. We're pretty familiar about mechanical transmission. It primarily happens through contaminated tools and sap transfer from person to plant. And so sanitation is really going to be your best measure for controlling these types of viruses. And these viruses that are mechanically transferred are extremely stable outside of the host. So you're going to have to follow through with sanitizing your tools, your benches, and your equipment. Also roguing infected plants, disposing of debris. Not touching healthy plants after handling infected ones. So if you do that, you're going to want to make sure you wash your hands or change your gloves out before you handle healthy plants. And then again, start with your virus-free propagation material. Due to the emergence of tomato brown gross fruit virus, protocols that have been traditionally in place for viral management are now starting to be reevaluated. So this is a virus that is mechanically transmitted via seeds, clothes, equipment, and irrigation, and it's also one that is extremely stable outside of the host. With respect to ornamentals, Petunia hybrida is an asymptomatic host, but to date, there hasn't been any symptomatic ornamentals. This is a virus that has been detected in Santa Barbara in greenhouses growing tomato. So in 2021, Chandra et al. Evaluated 16 disinfectants to look at the deactivation of tomato brown rugose fruit virus, looking at short exposure times. And then just to give you a little context about this study, this was not like a mechanical transfer study. This was evaluating deactivation. So treating virus particles directly with the disinfectant and then trying to infect a host plant. And so overall, what they found from this study was that a 0.5% and a 2% Viricid, 10% Clorox bleach, and a 0.5% lactoferrin were the most effective at reducing infectivity, although other products like Lysol at 50%, Vircon at 2%, and Virex at 2% also were able to reduce the percent infection. One interesting note about lactoferrin. So lactoferrin, for those of you who are not familiar, is actually a milk protein. Milk proteins have historically been shown to be able to deactivate virus. However, it's not recommended for greenhouse use. There are no registered products that use lactoferrin as an active ingredient. And the mechanism of lactoferrin for deactivating virus is actually not even known yet. Not only that, but because these are milk proteins, they do leave like a residue on your surfaces, which can be a little problematic. To give you a little more insight about those other products, so Virex is a quaternary ammonium-based compound, whereas Viricid is a QAB with glutaldehyde and isopropanol. And then Virkon XS is a potassium peroxymonosulfate product. Chandra et al. Did evaluate some ethanol-based products like Purell. However, they found that they were not effective at deactivating the virus, primarily because these are viruses that don't have that lipid envelope. And so the way that these ethanol-based products work is that they actually dissolve that liquid envelope, compromising the integrity of the capsid and the genetic material. So regardless of the products that you're going to use for sanitizing greenhouses, equipment, just to reiterate, Anna and Johanna, it's important to follow those product instructions, making sure you clean equipment prior to use since organics can reduce effectiveness. And then unfortunately for propagation tools, there's not really a lot of good options. The UCIPM website recommends bleaching your propagation tools in a 1-part bleach to 4 parts water. However, these tools must be kept wet for 5 minutes, and then again, rinsing well in water to prevent phytotoxicity. Just before I end this talk, I wanted to redirect everybody to the UCIPM webpage on viruses and viroid diseases. The link is down at the bottom of the slide deck, but it is just kind of fascinating to me, the host range of all of these viruses. And so it might be helpful to kind of understand what type of alternate hosts there are for viruses that might be impacting your grow. So with that, I have nothing else to share, but please feel free to reach out to me with any questions if you want to discuss further.
Thank you all for those wonderful presentations. Now, if the audience would like to unmute and ask questions, raise your hand, put things in the chat, please feel free to ask your advisor.
I think there are some questions in the chat that maybe we can answer.
Sorry, what did you say, Anna? My audio didn't come through.
There are some questions in the chat.
Yes. Okay. Did we go back and show that slide of the sterilization products again? Yeah. Okay, great.
I can also answer like the boiling water for how long. I would say always depend on which kind of pathogen you think you have, but I would say something between 15 minutes and 30 minutes will be ideal. Yeah, I can go to share the slide again.
Yeah, and then to add a comment on what Anna suggested, so like when we have heat treatments, in this case, boiling water, what we recommend is that it should be a minimum of 30 minutes, but it should be continuous. And so it's like not only like at one point it reaches, well, in this case, boiling water would be like 100 centigrades, but it should be kept during continuous 30 minutes. And like what we have when we're doing temperature treatments, the recommendations that we have is 60 centigrades at continuous 30 minutes to achieve the disinfestation or killing of pathogenic microbes.
So here is a slide of some of the disinfectant agents that are labeled to use in California. Some of them you will have to get into the label further because some of them may be authorized to be used in foot bath. Other ones may be authorized for tools. Other ones may be authorized for cleaning the surfaces. So you have to go deeper into the label to be sure you are using them for the purpose that you wanted. I think the next question is for Johanna.
Yeah, so there is no WPS label on chlorine used in nurseries. Are you recommending PW2, also known as Clean Grow? So Clean Grow is a quaternary ammonium. Clean Grow is a mix of quaternary ammonium and chloride. What I mean by chlorine is basically sodium hypochlorite, which is bleach. Yeah, and then there's also a chlorine gas that, for instance, that some cases is used for like treat the water. There's calcium hypochlorite or chlorine dioxide that can be used. Other questions is, are there organically certified sources of quaternary ammonium? I actually did like a quick search and there's not label products of quaternary ammonium that are organically certified. This is me doing a quick search on Google just when I saw this question, but I cannot speak about that. One thing of the quaternary ammoniums and the organic use is that they may stay for longer in the surfaces, so that may be an effect for organic production, but I cannot really speak about that. I don't know if anybody here, if Anna or Lydia know anything about that. And then next question, I think that's probably for Anna, but we can all answer. What is the safest way to compost plant waste from greenhouse if possible?
So I will say always bring them as far as possible from your facility, but I understand that that's expensive. So it will always depend on what do you want it to do with the compost. Are you going to finally going to trash that all material? If you are going to trash it, it's better to trash it as soon as possible. If you are going to reuse this pile of like all soil, if you wanted to reuse it, I will be more careful. Besides, like put it as far as possible. It will be good because sometimes we have crop debris in those piles, and fungi can or viruses can still be alive there. So it could be movement through the air to your facility or through insects from to your facility. So as far as possible, that there is no like wind currents that goes to your place, and also I don't know if you can put it in a container and try to measure. It's hard when you have like a huge facility to put it in a closed container, but at least you can measure the temperature inside to also be monitoring the temperatures that it gets and for how long, because the temperature and the time will tell you if it's killing fungi, bacteria, or weeds. So in order to reuse that again. I don't know if Lydia or Johanna have something to add.
Good. Moving with the next question, when sanitizing benches, how long should they remain wet and exposed to a sanitizing agent in order for products to be effective against bacteria, fungi, water molds? So recommendation is read the label of the product that you're using because it may change depending on the label. So for instance, like 5 and 20, which is a quaternary ammonium, the label says like 10 minutes. Bleach, in theory, has immediate action against any microbes. And so definitely read the label of the product that you are using. Another question, I think it's for Lydia. Can scales be a vector in theory?
I would say in theory, yes, but they're not exceptionally problematic or considered huge vectors of virus. There are a couple reasons for that with respect to their life cycle. There's only one stage in the life cycle of scales where they really move around a lot, and that's during the nymph stage. So, and I don't even know if they're feeding on plants. I think if I remember back to entomology, and I'll have to look into that again, I think most of the feeding occurs once they form that shell. And once they're kind of underneath that scale or armor, they're not moving around much. So they're not really considered, a significant vector.
Okay, another question we had is my ag department won't let me use Clorox at my nursery because there is no worker protection standards label for nurseries. Wow. I haven't heard of that. I don't know if people in other countries have had the same. In this case, what I would say, there are other products, for instance, such as quaternary ammoniums that are labeled to be used in nursery, and then that could be used instead. More question. Is there any recommendations for sanitizing plastic workspaces and benches? Sanitizing plastic workspaces and benches. Yeah, definitely. So what we recommend is if there are small surfaces, I'm part of the program Accreditation to Improve Restoration. This is a program directed to small scale restoration nurseries. And what we recommend for small surfaces is using 70% ethanol. That could be a spray on benches before doing any type of work. Also, having a chlorine or bleach spray of those surfaces is recommended. Other question, Johanna, did your study sterilize wood benches? Was the chlorine effective on wood benches? So no, we didn't do any studies with wood benches per se. What we did from wood is like from what we sample at the commercial nurseries. And so like we happened to find benches made of wood. And then at the time we sampled, this nursery didn't apply any treatment to the benches. And it's like taking a picture of the microbes we collected then. And so we did not apply any sanitizer to wood benches. Then when we did the study here at UC Davis looking the efficacy of different sanitizers, we did it on metal benches. The reason for that is based on the work we collected from the commercial nurseries that metal benches harbor lowered microbial loads, and then that is what we have also at UC Davis. And so we don't recommend having wood as benches. As I mentioned, in wood materials, microbes can overwinter, and they can be a source of harbor effort for inoculum. Other questions, apart from overhead watering, increasing air circulation, and sanitation practices, are there any other tips to minimize bacterial foliar diseases like bacterial leaf spot?
I will say increasing that avoiding overhead watering is great. The air circulation, the sanitation practices, all of them are super good. You have to keep doing that. But if these still persist, maybe you can try some bactericides, some products that are preventative for bacterial diseases too. If you are still having problems with bacteria, I will try those too.
There's more question. We use plastic nursery benches. They're lighter in weight, more mobile, UV treated. We still sanitize those, but not sure what the effect is on the surface of the table chemically when using bleach. We'll break it down. I have to say I'm not sure like what is the consistency of the plastic, but if you're concerned about that, I would say start by treating it with bleach in a small area to see if there's an effect of affecting or degrading the plastic. Also, as I mentioned, quaternary ammonium products could be used if there's a worry of corroding or breaking the materials that you have in your benches. This question, Lydia, I think it's for you, is Byron's question. How much should we concern about plant virus in terms of nursery sanitation? Are most plant virus pathogenic? It looked to me that plant virus are less problematic than bacteria and fungal disease.
Yeah, so I guess I would say that the reason to worry about plant virus is that with the exception of sanitation and insect management and weed control, there's very little once your plants are affected that there's just no control. They're affected and they have to be rooted and thrown out. The one thing about, and it also I think is dependent upon your production system, right? So if you're growing in a greenhouse, I think that you have a lot more opportunity for exclusion because, one of the biggest ways for plant virus to enter into your production is through, your propagation material. But you have the opportunity a better opportunity, I think, for keeping your greenhouses clean, eliminating pests and vectors. And so you have, I think, at least a closed system to prevent transmission. I think it gets a little bit more complicated when we're talking about outdoor production where you have a lot of weeds, you have a lot of, you can't necessarily exclude your insects from coming in. And if you look at that list of plant virus, they're diverse. They have a huge host range, some of them. And so I think it's going to be a lot more of an issue and, for sanitation and weed management and just being vigilant about looking for those symptoms and roguing.
Good. And another question we have, does anyone have a list of items they should keep in their sanitation box in the greenhouse? That's a great question. And so I will start by referring people to the Accreditation to Improve Restoration website that we have. So what is Accreditation to Improve Restoration? Is a program that we have for restoration nurseries where we have strict management practices to exclude the presence of Phytophthora to prevent outbreaks into native ecosystems. In there, we have very strict sanitation guidelines, and there are some guidance of sanitation practices that should take place. I think I posted the website to everyone there, but in general, what should be on the sanitation box? I would say first, something to clean. So any type of brush that can physically remove plant debris, organic matter, or soil. Then I would say having the sanitizer that take place in your nursery, given that is chlorine treatment or quaternary ammonium treatment, making sure you have the label handy to prepare the adequate solutions and concentrations that they should be done. And then the more tools depending on the part you need to sanitize. For instance, like you'll need clean gloves, you will need clean tools, you will need clean footwear. And so depending where you are working on the greenhouse, but I would say something to physically remove any plant debris or any organic matter, and a sanitizer agent will be like the main components in your toolbox for sanitizers or for a sanitation box.
I added some bactericide products that for Gabriela Gallardo that she can also use as part of the hair IPM strategy for bacterial foliar diseases.
Okay. Any other questions for any of us?
Well, I guess there's about IPM. How can I establish an economic threshold for thrips in Ficus benjamin ficus plants before reaching a population density that increases risk of disease transmission and sacrificing crop quality? So I think my recommendation for economic threshold and thrips is that, I think it's important to remember that, the thrips are not necessarily carrying the virus. They just have the capacity to vector it. So if it's in the greenhouse, that's when. So I think in this case, I would start thinking about like the economic threshold, not for thrips potentially carrying disease, but just for your thrips in general. I mean, just it's always best to keep your pest insect populations as low as possible, and just kind of keeping in mind that the virus is just an added risk that could be occurring in the greenhouse. But if you've already got it established for thrips in general for your production, then you could remain with that.
Okay, it's 11 a.m. Unless there are any more questions, I think we may be ready to wrap up.
Thank you, everyone.
Yes, thank you so much for coming. Thank you to our presenters.